The foundation of bacteriology as an experimental science was the development in the 19th century of cultural techniques using solid or liquid media. Culture allowed viable bacteria to be detected and isolated, facilitated the observation of metabolic activity, and provided biomass for further analysis. The most commonly used forms of bacteriological analysis in food microbiology are detection and enumeration. Detection of specific types of bacteria can be achieved by cultural isolation or by indicators such as biomolecule specific to the organism or products of metabolisms. Platforms developed for healthcare cannot be easily adopted for use in food microbiology as an analysis is significantly more challenging. Analysis for the presence of bacteria that cause foodborne intoxication, spoilage or serve as production aids does not generally require limits of detection below 100 ml. Culture-independent diagnostic platforms for infectious agents have been successfully commercialized in the healthcare sector, and the potential for speed and automation has stimulated interest in the application of similar systems for food analysis. Culture media are used for quality control tests of non-sterile raw materials and finished products, as well as for microbial contamination tests in applications such as hygiene monitoring, sterilization process, validation, and determination of the effectiveness of preservative and antimicrobial agents.
Next is about predict the bacterial phenotype. So the complementary nature of genomic and phenotypic data is apparent when attempting to understand and predict bacterial behavior. There's actually a wide range of bacterial behavior of interest to the food microbiologists. Genomic analysis also allows the researchers to rapidly detect known genes, putative genes, and other defined features of genome. It is also used to screen large populations for strains that potentially possess desired phenotypes. So next is the purpose of the mutation genomics is actually not to imply that the application of genomics to answer these questions is inappropriate. Decision-making related to food safety should not be made solely on the basis of genomic data, but it also needs to be supported by phenotypic data, which in turn require culture. When grounded in phenotypic data, genomic data has the potential to enhance culture methods or to develop culture method for previously unculturable organisms. And next is genomic technology. It actually can be defined as tools with enormous potential for increasing our understanding of bacteria and solving practical problems in food microbiology. Culture also remains necessary to achieve the required sensitivity of detection and enumeration, and also to determine viability. Next, I will talk about WGS data. The application of WGS data for isolate identification, subtyping, and source tracking in the context of public health, regulatory, and also commercial decision making is very recent, and standards for analysis and interpretation have yet to be established.
Moving on to immunoassay, starting with the principles of immunoassay. Immunoassay use the natural mechanisms of living system to develop particular antibody that interact with foreign molecules. The introduction of antigens into a live organism causes lymphoid tissue to produce antibody in response to antigen recognition. The specificity of antigen-antibody interaction, also known as immunological reaction, is extremely used for clinical use in infection disease diagnostic and therapeutic drug monitoring, as well as more recently for identifying and quantifying food consistent agriculture chemical and microbiological products. For immunological methods, in immunoassay, food chemical constituents and microbial toxins or cellular structure are injected in their pure form or in conjunction to a protein such as bovine and serum albumin. This is followed by screening of those clones that secrete antibody of predefined specificity. There are two types of immunological reaction observed, which is the first one is precipitation occur when soluble dietary ingredient of microbial quotient react with particular antibodies. Following response with their respective particular antibody, cellular antigens including pathogenic bacteria, viruses, and treated red blood cells are glutinate. The identification of the antigen is determined by the specificity of immunological reaction. The advantages of this method is the first one, this method allows increased sampling and rapid movement of food between production and processing, thus further reducing spoilage and quality losses. Next, it can reduce requirements of chemical and sophisticated equipment and also help bring down the assay cost by up to 90%. Last but not least, the reduction in assay time to a few hours or less per estimation as compared to several days for conventional analysis has allowed for increased production.
The immunoassay technique is commonly used in food industry including food production, food processing, and food safety. Various immunoassay systems such as enzyme-linked immunoassay (ELISA) and radioimmunoassay (RIA) are used in detecting microorganisms in food that can cause foodborne illness. The early detection of viruses such as viruses in the seed helps to avoid food losses as viral infections tends to spoil the food crops. For instance, the immunodiffusion method is used to detect elongated potato virus in potato. Other than that, RIA is also used to detect the cauliflower mosaic virus in the cauliflowers. These immunoassay methods can be applied in the research of the virus's taxonomic status that can infect the various crops. The use of ELISA to determine mold counts helps to prevent food spoilage, as this method can estimate viable and non-viable organisms through reaction with antigenic polysaccharide. ELISA method can be applied for mold counting in tomato puree, and the test showed that ELISA method was 100-fold more sensitive than HPLC method for counting molds in tomato puree. Immunoassays are highly used as diagnostic tools to detect the spoilage microorganism in foods. For example, ELISA method is used in soybean to detect Phytophthora megasperma that naturally infected soybean plants. Various foodborne fungi are able to identify by examining the morphological characteristic of the spores and also the mycelia using light microscopy. Thus, immunoassay can be used in determining the bacteria and also mold in the food efficiently.
Polymerase chain reaction is a method of in situ DNA replication that allows for the exponential amplification of target DNA in the appearance of synthetic oligonucleotide primers and thermostable DNA polymerase. To detect bacterial isolates, a PCR process may employ one primer or multiple primers. Real-time PCR, nested PCR, reverse transcription PCR, and many other types of PCR exist. Polymerase chain reaction assays have long been used to detect, identify, and differentiate foodborne pathogens. The polymerase chain reaction technique is useful to identifying typical bacterial strains that exist in viable but non-culturable cocoid form and are frequently missed by the conventional method. However, because amplification can be obtained from both viable and non-viable cells, some PCR may not be suitable for process of certain food. The technique can be costly and its sensitivity and performance can be compromised by enrichment broth and DNA extraction solution components, PCR mixture concentrations, and temperature and cycling conditions. Next, single polymerase chain reaction. This is polymerase chain reaction that detects an organism by using a single primer set which targets a specific gene. The primer set can be tailored to a specific species and can detect the target organism even in the presence of other organisms. This type of PCR can be used to detect or identify bacteria in a food sample quickly with or without pre-enrichment. Single-step PCR can also be used to confirm bacteria isolates obtained directly from agar plates. PCR with universal or specific primers has recently been used to amplify the 16S rRNA genes of bacteria before sequencing to aid in the identification of unknown or novel bacteria species. This is polymerase chain reaction that detects an organism by using a single primer set which targets a specific gene. In 2011, Rahimi et al. Used PCR to confirm the identity of 39 Campylobacter species isolated from 110 duck meat samples in Iran using the traditional bacteriological method. ZT et al. 2012 used PCR to amplify Campylobacter species 16X rRNA before sequencing for species identification. This PCR can detect single bacteria or genes more accurately, sensitively, and quickly, but does not yield isolates that can be further characterized. Food components can interfere with PCR performance and produce false results. PCR conditions must be optimized for better performance.
Multiplex polymerase chain reaction is a modification of polymerase chain reaction that use multiple primers within a single PCR mixture to detect, identify, or differentiate bacteria. Thus, in multiplex PCR, more than one target sequence are amplified in a reaction to produce amplicons of varying size specific for different DNA sequence. The advantages of multiplex PCR are reduced cost, limits volume of samples, and allow for the rapid detection of multiple bacteria species. Other PCR that can be used is real-time PCR. Real-time PCR employs specific primer set 1 or 2 probes to improve detection signals. In real-time PCR, the amplified DNA is detected in real time as the reaction progresses instead of at the reaction end. Real-time PCR shortens detection time compared to standard PCR and can determine the absolute or relative number of bacteria in various samples. Next is reverse transcription PCR. In reverse transcription PCR, RNA is used as the initial templates instead of DNA. Reverse transcriptase is used to reverse transcribe the target RNA into DNA complement and amplify using PCR. Reverse transcription PCR is useful in detecting only viable cells of pathogens. However, RNA is unstable, requiring much skill during handling and quantification for pathogens. Lastly is nested PCR. Nested PCR employs two sets of primers in two successive polymerase chain reaction runs in which the first PCR product generated is used as primer for the second PCR. Nested PCR improves the sensitivity and specificity of detecting foodborne pathogens compared to the conventional PCR.
I think that's all from us. Thank you for watching.